In this first part of the lecture, we will discuss the genetic phenomenon of codominance using two examples from human biology, both related to blood typing. Codominance is the term applied to the situation when two alleles of a single gene are responsible for producing two distinct, detectable versions of the gene product they encode. In a heterozygote, this results in a phenotype that is not one or the other, but both possibilities simultaneously. We will first consider the MN blood group in humans, and then the ABO blood group system, also in humans. Before we can do that, however, we need to talk about antigens and antibodies a little bit. An antigen is a cell surface structure, usually a glycoprotein, that identifies the cell. It is called an antigen because it triggers antibody generation. So antigen. The anti part is antibody and the gen part is for generation. This little pale blue cell over here is depicting something like a bacterium. And it has projections coming off its surfaces, so here, here, and so on. And these are representing antigens. Okay. Over here, we have a B cell, one of the types of human cells that make up our immune system. And the B cell is producing antibodies. These Y-like structures. These are protein structures. And you'll note that the antibody very specifically binds to the antigen. For each particular antigen, the B cell is capable of producing an incredibly specific antibody. When that binding occurs, what will happen is you'll get massive binding, okay, and it'll essentially you'll get this clumping of antigen-antibody binding all over the surface of this little cell, or foreign particle if it wasn't a cell, and that essentially targets the cell for removal. So binding is going to equal phagocytosis. Cells like macrophage are then going to recognize that they should clear or remove that particular particle or invading cell. In this way, we keep our body free of foreign particles, such as invading bacteria or viruses. And that's the basics about antigens and antibodies. So this antigen-antibody story has a lot of relevance to many aspects of our biology. For example, how we protect against pathogens, but also relevance to the issue of tissue transplants. It is the underlying reason why you must only receive tissue from an individual or donor who is an identical match to you when it comes to the genes for encoding antigens. This includes blood transplants. So in scenario A, shown on the top of this figure, the donor blood over here, okay, the donor blood has antigens on it, and these little round circles are depicting the antigens, okay. In this part of the figure, we're seeing the antibodies that are present in the receiver blood, the person who's getting the blood. And what we'll note is that the antibodies are not binding to the antigens. So we say that no agglutination reaction has occurred. And that's a good thing because it means that the red blood cells then are free to do the work that red blood cells are supposed to work, and the recipient or patient will be healthy, the blood transfusion will be successful. Okay, so what that tells us when I say, what do we now know about the receiver? It tells us that there was a really good genetic match between the receiver and the donor, because we don't make antibodies typically that will bind with our own antigens. So the antibodies in the receiver's blood are not recognizing the antigens in the donor blood. Okay, so that means that probably those antigens are very similar or identical to what the receiver has on their own normal red blood cells. Now it's a different scenario below. In scenario B, we've got donor blood, and we have some different antigens showing on the surface. And then here we've got the receiver, the patient's antibodies, and we're seeing a match. Okay, so the antibodies are binding the antigens. That's what's shown here. And the result of that is clumping or agglutination, and that targets these red blood cells for destruction, which is called hemolysis in the case of red blood cells. And that's a grave problem for this patient because the red blood cells are no longer able to do what they're supposed to do. They can't transport oxygen, and they're also probably going to go into, they're going to have massive clumping in their circulatory system, and that can be lethal. So that's a grave problem for this recipient, often causes mortality or death. One of the antigens on the surface of our human red blood cells is the MN antigen. There are actually 3 alleles for the gene that encodes that antigen, but the most common are the LM and the LN that are shown in this figure. So you can have genotype LMLM, LMLN, or LNLN. And the resulting phenotype is also shown here. If you are a homozygote, your phenotype is fairly straightforward. If you're LMLM, then your phenotype M, and you only make the M-type antigen on your red blood cells. So let's just pretend, let's draw a red blood cell down here. Let's say that you're LM, and you make M-type antigens on the surface of your red blood cell. Similarly, if you're a homozygote for the N allele, you'll also make, you'll express those genes, but your antigens will look a little bit different. They might look like this. Okay. Now what's interesting is that in the heterozygote, LM, LN, they are going to show us what codominance really means. Their red blood cells are going to contain both M-type and N-type. Getting a bit crowded here, okay? It's not that they make more antigens, it's just that they make both forms. Okay, so they have both types of antigen. So this is a case where we're seeing both the M-type and the N-type simultaneously, both versions of the gene product that are possible here, and that is what codominance means. This is a classic illustration of codominance. So blood grouping is more complex than just the examples that we're looking at, but let's consider another type of blood grouping or another blood group system, the EBO blood group system. This was discovered by this guy whose name was, Carl Landsteiner. Okay, and it was discovered early in the 1900s. He figured this system out because he was interested in trying to understand why some blood transfusions saved people's lives and others actually killed them. And so the ABO, or blood grouping system, means that we can be blood type A, B, O, or AB. And these are the results of genotypes. These are the results of antigens that are formed from genes that are just referred to as the I gene. The possible alleles for the I gene, there are 3. They are IA, IB, or just I. And let's consider the biology of this system now. There are 6 possible genotypes with regards to the I gene or the I locus, and they are IA, 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 I, IB, 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 I, IA, IB, and I. Okay? If you are IA, then the antigen that you have present on your the surface of your red blood cells is the A antigen. Okay? And the blood grouping or phenotype that you belong to is also A. You are type A. If you make IAI, you also are phenotype A, and you make antigen A. Okay? Your antigen determines your phenotype. Okay? It dictates. If you're IB or IBI, you make the B type antigen, and your phenotype is therefore B. If you are IAIB, you make both A and B antigens, and your phenotype or your blood typing is type AB. If you are just II, then you make what's called the O antigen, and your phenotype is, actually you make neither antigen, and your phenotype is considered type O. Okay? All right, let's look at a sort of a cartoon representation of this to hopefully make it clearer. An important point to make here is that we do not typically make antibodies, which are shown in the bottom part of the figure, that are against or that will bind to our own antigens. That's a really important thing that you need to remember in order to understand this biological story. So let's take a look. This is a cartoon that illustrates some of the principles that I'm talking about. Okay? If you are a type A individual, which means your genotype is IAIA or IAI, then it means that you make A-type antibodies on the surface of your red blood cell. So you're not going to make antibodies that are against A. Instead, you're going to make antibodies that are against B, the other option. Okay? And that's what's shown here. Okay? Similarly, if you are a type B blood type, it means your genotype is IBIB or IBI. You will make B-type antigens on the surface of your red blood cells. That's what's shown here. Okay? And you will make anti-A antibodies, which is what's shown down here. Okay? If you are type AB, then you're the codominant. You're showing that particular phenomenon. So you're making both A and B type antigens. So that means that you cannot make anti-A or anti-B antibodies. So you make no antibodies in this regard. You of course make other kinds of antibodies in your system, but not antibodies against A or B antigens, because then you would have clumping in your own blood supply. If you are type O, that means your genotype is II, you don't put any A or B antigens on the surface of your red blood cell. You've got this little naked looking red blood cell here. So that means that you will make both anti-A and anti-B antibodies in your system. Let's think about the implications of that from the point of view of blood transfers. This figure shows the result you would expect if you actually did an experiment. You could do that by mixing two blood types on a microscope slide and just looking with the microscope to see what would happen. But it shows what would happen if you did that experiment and you gave donor blood to recipients of different blood types. Okay? So the top row shows the donor identity, O, A, B, or AB. You should be able to draw their possible genotypes at this point. The left-hand column shows blood, like what the recipient's blood is. Okay? Again, O, A, B, or AB. Clumping means agglutination. So in this reaction over here, for example, sorry about that, we're seeing clumping, which is agglutination, which is due to an antigen-antibody binding reaction. Okay? over here is an example of a figure where there's no clumping. So we're not seeing any kind of antigen-antibody binding in that case. You'll note that the O person is able to successfully give blood to anyone without clumping occurring. That's shown through this row. This is why we call O the universal donor. In contrast, however, the type O recipient here can only get blood from another O. They can't get blood from an A, a B, or an AB. Okay, so they're a little bit more limited in their donor possibilities. Interestingly, the type AB can receive blood from an O, A, B, or an AB. So they're fine no matter where they get their blood from. Type AB is therefore called the universal recipient. The ABO gene product, or the I gene product, is an enzyme that is a glycosyl transferase. Transferase always means enzyme that moves something. Glyco tells us sugar. This is essentially an enzyme that can move sugar groups from one place to another. This particular enzyme can add sugar groups to pre-existing sugar structures in a glycoprotein antigen. Here, the pre-existing polysaccharide is called the H antigen. So this is kind of like the building block. So we would have like the surface of the red blood cell, and then we would just have these little antigens poking out, and they are the H antigen. Okay. The glycosyl transferase enzyme encoded by the I gene can come in two possible forms. The form that is the result of expressing the IA allele, or the form that is the result of expressing the IB allele. If you're expressing the IA allele, then you make a form that is capable of transferring this sugar, okay? These are both sugars, onto the H antigen. So you might get a red blood cell that now had antigens that looked like that, because the H portion contained a structure that the shorthand is NAG, okay, or alpha-N-acetylgalactosamine. If you express the IB allele, then you make a glycosyl transferase. It's a little bit different. Its active site will be different, and instead of transferring NAG, it will bind to galactose, and it will place it onto the H antigen. So in that case, you might get a red blood cell. You would get a red blood cell that had a different antigen, the B type. So we could draw that differently. It might look like this. And of course, if you were AB, it means that you are making both forms of that glycosyl transferase enzyme. And so you would get a molecule, you would get a red blood cell, that would have both this type antigen and this type antigen on the surface. That's the molecular basis underlying this ABO blood system. So the I gene product is glycosyl transferase, a particular glycosyl transferase. Here are some examples of questions that I would consider fair game for a quiz or an exam based on this lecture and the readings, the accompanying readings in your textbook. So please go through these questions and note the level of detail required. Just notice there's some screw-ups in this thing, so this should be, this should be in your answer, include the terms antigen, antibody, and agglutination, the 3 A words that matter here. And in this example, question number 3, make sure that you could answer this not just with yes or no's, which doesn't actually show that you have any understanding of the system whatsoever, but that you can explain your logic by writing out possible genotypes for parents, and possible children. I'm not going to spend any time on the Bombay phenotype. There is a question in your mastering homework that addresses this, and it's well explained in your textbook. Okay. Please move on to the next part of the lecture now.